to about 380 participants. They're increasing numbers, but I want to let everybody know that this town hall will be recorded, and that the Q&A that you use to ask any questions we will keep and we will use as a FAQ format for future, but we will be addressing as many as we can today. So I'm Kelsey Martin. I'm the Dean of the David Geffen School of Medicine. I want to welcome all of you. I want to thank our entire research community for the incredible effort that has been put into addressing COVID-19 in the most collaborative and impactful manner possible. We have put together, in response to the COVID-19 pandemic, an overarching organization for our research activities. This is aimed at ensuring that we do, again, the most impactful work in the most responsible manner, and that we contribute to the solutions to COVID-19. Along these lines, we've set into place, as you saw in an email that came out about a week and a half ago and another email a couple days ago, a series of structures that allow us to really attend to how do we come together, how do we leverage the expertise of our community so that we can utilize the information that's available on COVID-19 in patient populations and healthcare workers, again, responsibly and impactfully. The real hope here is that the solutions that are coming out of our research efforts are going to be the solutions that are going to be bringing our society back to a normal functioning state, that are really going to address all of the healthcare needs of patients with COVID-19. So with that, I'm going to turn it over to Dr. Arash Naim, who I think may still be getting on the call because he looks like he got bumped off of the Zoom town hall. One of the amazing things that we've come together so effectively using IT, but technology, but every once in a while we get some problems with it. So I will just use the time to just talk a little bit more about some of the overarching activities. I think that you'll be hearing about amazing work that's being done in the clinical trials category, really bringing a series of clinical trials to UCLA Health and their patients in record time. You're going to hear about research in translational to clinical research that promises to have just amazing impact. And with that, I now see that Dr. Naim is back on, and so I'm going to turn, no, you can't hear anything. Is that correct? You can't hear anything, but everybody else can hear, right?
Yeah.
Yes. Okay. So Arash, he probably can't hear me. I was going to, I'm going to text him and tell him to call in with his telephone. But he is starting to share screen. So maybe I can pass this over to one of our panelists. We have a series of panelists who are here today. Steve Dubinet, the head of the CTSI, Judy Currier, the chief of infectious disease, Steve Smale, the vice dean for research, Sarah Dry, the interim chair of pathology and laboratory medicine, David Ellishoff, who runs the biostatistics core in the CTSI, John Del Perio from pulmonology, John Glaspie from Hem-Onc and the JCCC, incredibly important partners. As you know, most of our clinical trials historically have been in the area of oncology, so huge expertise and partnership there. Again, Arash Naim has also joined us. And with that, I'm going to turn it over to see if one of our panelists can then start going through the slides.
Hi, this is Judy Currier. Can you hear me okay?
Yes.
Okay, so until Arash can get on, I can start us off and just to provide an overview. And I want to thank everybody for joining today, and also acknowledge Arash's leadership in this COVID research task force activity. And the group is really trying to make sure, as Dean Martin has said, that we prioritize the most impactful research that can feasibly and quickly be conducted. And so the task force has been put together, and there are many components of this activity. On this slide, you see the main scientific prioritization committee, which is chaired by John Belperio, and has others representing different fields of epidemiology, health services research, basic and translational clinical research. And this group is reviewing proposals that come in to use data, specimens, and other resources of the health system to study COVID. And they are meeting regularly to review proposals. Arash, are you there now?
I am on. Can you hear me?
Yeah, I hear the echo because I think your computer and your phone are both on.
All right. Are you good now? Can we hear?
Yeah.
Much better.
Okay.
Much better.
Sorry about that.
All
Okay.
right.
So, sorry about that. So as Judy was saying, I think with the leadership of the dean's office, they drafted individuals that I think are very committed. I will say that there are over 140 COVID-related human subjects applications in. So the work that this group has to do to be quick in terms of doing a quick review, I think is important. And their charge largely is to focus on studies that either access our UCLA health patients, with an emphasis on the inpatient side being the biggest concern, access to UCLA healthcare providers, many of whom are patients, but many of whom are not and are managed through occupational health, clinical data that's derived from those populations, specimens that are derived from those populations, whether they're clinical remnant or whether people are going into the rooms to consent them for research collected specimens, and treatment studies for those cohorts of individuals. And the reason that the leadership created a committee is that there's an increasing number of competing and overlapping studies being submitted to the IRB. The IRB really has been a great partner here in reviewing these very quickly, but they're also having a difficult time sorting some of the applications that are coming in. The desire to maintain the privacy of our patients, many of whom are also UCLA employees, the limitation on personal protective equipment, the fact that there is significantly reduced operational resources during the crisis, and the definitive limited biospecimen availability while people are in the hospital specifically. So the committee is focused in really three main areas. One is clinical trials, one is related to biospecimens, and the other related to how we can provision data as effectively as possible. So with the trials, I want to pass it on to Judy so she can give an update.
Okay, thanks, Arash. Clinical trials evaluating promising therapies has been a really high priority for the group. And we look at this across the spectrum, from prevention, post-exposure prophylaxis, early disease, to inpatient trials for moderate to significant serious disease. And if you look over on the right side for the inpatient trials, this is where we've really had the most activity, the quickest. And I think this is important given that we have no known effective treatment for this disease. So shown in red are the studies that are currently open and enrolling at UCLA, Ronald Reagan, and Santa Monica hospitals. And these are evaluating a range of interventions from antivirals with remdesivir to anti-inflammatory IL-6 inhibitors to other immune modulators as well, and other agents. And the studies in yellow represent interventions with a variety of different mechanisms of action that are in the process of being opened. So that you can see that in less than a month, very quickly, with the help and incredible expertise in the CTRC and through our contracting office, and got multiple studies open. These are being led by investigators in infectious diseases, Otto Yang, and pulmonary, Stephen Chang, Tisha Wang, and others. We're also in the process of getting compassionate plasma for treatment, and expanded access is started to enroll donors, and hopefully we'll have randomized trials to evaluate the efficacy of that intervention. Moving to the outpatient setting, we do have studies that are about to open for outpatient treatment. None of these are currently open. And then on the left, this one study that did just open this week is a study looking at hydroxychloroquine versus placebo as post-exposure prophylaxis. And this is an intervention for people who've been exposed to a confirmed COVID case. So this could be family members or household contacts of healthcare workers or others who've been exposed to a case. And that's funded by the Gates Foundation and is just started to enroll. So you can see that, I think really quickly a lot has happened. Go on to the next slide. But what's also become very clear is that multiple efforts to study treatments overall from sort of a national perspective have not been well coordinated. There's been certainly a lot of effort from industry and NIH and others to get these studies stood up. But what's been missing is a coordination. That has become, obviously an important area of focus across the NIH. And the next slide, I think will show that NIH has launched a public-private partnership through the NIH Foundation to try to speed the development both of vaccines and therapeutics for COVID. And they're planning to try to develop more integrated approach across industry and NIH to stand up studies that could potentially more quickly evaluate agents in master protocols with a common control arm. So really looking forward to more information coming from this group to help coordinate the effort moving forward.
Thanks, Judy.
That's on, back to you and biospecimens. Thank you.
So, of course, biospecimens are important as part of the clinical trial protocols themselves, but there's a lot of research that's being done by the translational teams that require biospecimens, especially in the areas of immunity and virology. And so the types of specimens that seems like there have been a lot of requests for include viral type of tissue, mainly coming from the nasopharyngeal swabs, but people are trying to collect a variety of other types of viral samples, and blood in terms of the serum and the plasma and the cells. And the strategy that we've tried to utilize to service the needs of the entire research community comes from infrastructure that was laid fairly early in this process. So pre-existing COVID, we had a remnant outpatient and inpatient approach to collect remnant clinical samples that was developed by the Clinical Translational Science Institute and Institute for Precision Health as part of the ATLAS program. And that has been largely utilized now to redirect the COVID specimens so that we can make them readily available. And in part due to the activities of Dr. Fulcher and the infectious disease doctors, a pathway to consent all of the inpatient COVID positive patients so that we can collect a robust number of both viral and or blood specimens on them during the course of their hospitalization has been created, the majority of which will be banked for researchers to be able to access. We are now seeing a lot of people that have recovered, and as they convalesce, the interest in being plasma donors as well as participating in research has increased. And just last week, when the call went out for recovered individuals that want to participate or donate plasma, over 1,000 people completed the form to be part of that registry within 3 or 4 days. And so when they come back in to be screened for potentially being a donor, additional research tubes are being taken so that they can support research efforts that are happening to follow the immunity of people that have had the disease and have recovered. Largely that's supported by the work that Alyssa Zyman and Otto Yang are doing as part of the convalescent plasma program. And then largely due to the leadership of Dr. Dry and the people in anatomic pathology, there's a pathway to get potentially some research consented autopsy specimens as well for studies that need that. And there has been at least 1 or 2 submissions that have had an interest in those tissues as well. And I think that this strategy largely reduces using these sort of infrastructures, the point-to-point sort of need for pathology and laboratory medicine to support the research community at a time when they're feeling very overwhelmed with the testing. But to speak to that a bit more, I wonder, Sarah Dry, if you can give a couple of words on what we're trying to do here. I don't know if Sarah
Yeah,
can
thanks.
unmute.
Oh, sorry, I just had to unmute myself. Sorry about that. Yeah, thank you, Arash, and good afternoon, everyone, or good morning, I guess still. Yeah, Arash, you summarized it very well. We have been very busy within pathology, focusing on clinical testing in order to ensure that we have adequate testing for all of our patients and healthcare providers. In terms of biospecimens, as both you and Judy noted, one of the concerns are that these are a finite resource. Another concern is that there are relatively few hospitalized COVID patients relative to the number of studies. And there was significant concern within pathology early on that without coordination, we could inadvertently end up harming our patients clinically because of all the biospecimen collection that was requested. And so the process that's being set up will help alleviate that and ensure that that doesn't happen and that our patients are protected. And then the final note that I just wanted to make is that there are resources that are required to collect these specimens, and it's important that we ensure that we are using those resources appropriately given all the other demands on the system at this time. So that's really all I wanted to say, but that we are very happy to support biospecimen collection and procurement to research projects.
Thanks, Sarah. And then that kind of leads to data. As many of you who have been accessing data have realized that there are multiple elements to being able to collect the data that might be important for this population. There's data that's in EPIC, people that get tested, the people that test positive, associated with those individuals, both inpatient and outpatient. There are a variety of very discrete elements in EPIC. We've negotiated a way that that information can be pushed into a CTSI REDCap environment where the PHI is available but locked so that it can't be downloaded in a way that satisfies our compliance and regulatory responsibilities. There are some information that is not discrete and requires chart abstraction in order to be able to reliably pull out. Some of those are part of the WHO core forum that many of the international and national collaborations require, as well as additional pieces of information that are pertinent to UCLA interests. And to ensure that the people that are going into the patient's chart that are abstracting these are a small group of people that compliance is aware of, because many of these individuals that are positive may be our own patient population, our own working population. A centralized chart abstraction team under the leadership of David Elishoff will be managing it so that there can be transparency about who's doing this. And so it's also transparent in audits for compliance as to who's accessing these charts. And then lastly, there are some activities that UCLA Health is doing as part of either quality improvement or population health. And that information can also be integrated in this infrastructure. And the person that is going to be responsible for this is Dr. Elishoff. But I will point out, before I pass it on to him, that if there are proposals that are focused primarily on COVID positive or COVID positive matched COVID negative controls, and they use this infrastructure that is managed by Dr. Elishoff and request only de-identified or limited data, meaning only dates or zip code, and they sign an internal data use agreement, or if they're collaborating, the receiving academic collaborator signs an external data use agreement, then we've arranged to fast track those through compliance, the data sharing release committee, and if it's an external third party, HDOC, which reviews all of the external third party agreements. So this may be the fastest way for people to get data and collaborate, whereas other sort of one-off requests for data may have to go through the regular process. And because we're short-staffed, that may create delays. And David, I wondered if you could speak to that a little bit.
Sure. Thank you, Arash, and good morning, everybody. So the basic design of the infrastructure is that we'll have a common data repository, as Arash was describing. But then for each individual research project, we plan to create an individual REDCap that will encapsulate the data elements that are needed for that particular research project. So for researchers interested in working with this data, we suggest that you reach out to the CTSI Biostatistics group. We provide consultations on database design. Importantly, we also want to work with researchers to identify what data elements they need and potentially add those to the UCLA core ECRF. So if there's additional data elements that are needed for a project, we want to make sure that we're collecting them in the uniform way to support the research projects. So I think that we have an intake form that can be filled out to describe the study population, and then subsequent to that, we have a data dictionary to describe all the data elements that we have currently in our collection protocol, and but we're adding to those all the time to provide support to research projects. So for the central abstraction, the idea is that we want to make sure that that multiple research projects are not trying to enter the same data on the same patients, and that we can really enter data in a consistent and high quality way.
I also think that, David, that also allows us to ensure that people are not setting up, there are many people that are involved either operationally or they're as part of the care process and may know at least what individual cohorts of COVID patients, what their medical record numbers are, or et cetera. But at least if the intention is to pursue some sort of a research project, being able to pursue the chart abstraction in this way allows a process that can be audited and reviewed in a very consistent way, so that it doesn't run into problems where people are accessing the data for one particular purpose, but then utilize it in some way that then is inconsistent with institutional policy. And I don't know if, Kelsey, you want to speak to that anymore, because I know that there's been a lot of discussions on that over the last 24 to 48 hours.
No, I think you covered most of it. I mean, it really is, a combination. We need to protect patient information, employee information, but we also, of course, want to learn what we can about the infection.
Great. So with that, maybe we can just see what the questions are that people have about the process that we're trying to set up. And I think that Steve Dubinet is going to manage that process.
I think that there were a couple of questions about the data centralization that have been answered in general. One question is whether the process described relates to non-COVID related studies. In other words, is this for observational studies and QI, is there a separate process?
Yeah, so I mean, I think that everyone realizes that the quality improvement efforts are mainly to serve our patient population and to improve the care in the hospital. So I don't think the goal of any of these processes are to in some way interfere with that. I do think that even those projects at some point or another need IRB review, and that there are business owners in the hospital that are responsible for those initiatives. And if data mining is going to happen as part of a quality improvement project or some other operational activity, the business owners for those should be aware of them, so that they're endorsing that sort of activity as part of the operational or QI process. I think that's the main idea around that.
Okay, and there's a couple of questions about the actual collection of research biospecimens and how the PPE is being organized for that.
Yeah, so to the extent possible.
Sarah, do you want to talk about that?
Sure, yeah, so within pathology, we have all of the appropriate equipment. We have negative pressure rooms. We have saws with appropriate containment structures. We have appropriate PPE. So there's no need for additional resources within pathology at this time.
I think they might have been asking about whether there are resources for other investigators who are using samples outside of the organized effort, maybe.
Oh, I'm sorry. I can't answer that question. That would have to go to someone else.
So for, and this is Steve Smale, for resources that are, needs for additional space, I think this is something that, if biospecimens requests go through the committee and are considered to be, high priority needs, and an investigator does not have the, the space or ability to do what they need to do, then it'd be good to work that through the department chair, who will bring it through the dean's office if necessary to figure that out. It's important to note that patient specimens, require BSL-2 biosafety containment. So there's an abundance of BSL-2 facilities around the campus.
And Dr. Smale, can you also speak to issues related to non-human research? There's a couple of questions about that and who the process.
Which questions are you referring to? Just so
the process for authorization for studies that do not involve human biospecimens.
Okay, so there's really not a, so for laboratories that are interested in doing COVID-19 research, that they, at this point, everyone requires an essential experiments approval, which is the same approval that was announced a few weeks ago and is listed on our website, where you need to just send an email to your department chair explaining what you want to do, describing the staff that will be involved, the amount of time, making sure that you have plans for social distancing when the work is being done. The chair will evaluate it and then send it to me for approval. So everybody that's doing laboratory research needs to have essential experiments approval. But other than that, there's no approval needed to be able to get those experiments off the ground. If anyone that wants to work with live virus, there's a need to go through a committee that's called the High Containment Use Committee, that's evaluating and prioritizing space within the high containment facility, because that facility is quite small and there's a lot of interest in using it at this point. Yeah, and again, if somebody needs a new resources such as BSL-2 hood access that they don't currently have, and they have a high priority project, that the department chair can often help solve that problem, but can come to us if they are unable to do so.
I want to just add one thing to that, which is that in addition to the request for permission to do essential research, we are starting, and it was in the initial email that we sent out on March 31st, a basic to translational research group. And the goal there really is, just as you've seen this incredible progress that's been made, for example, in clinical trials, because people have really come together to determine what are the highest impact. We believe that in the basic to translational arena, it would be very helpful for some levels of collaboration that could really address incredibly impactful questions in cell biology, structural biology, immunology, et cetera. So that will be coming out. We're rolling things out as the situation is evolving, but I want people to be aware of that as well.
Okay, David, there's a couple of questions that are specific relative to the REDCap. How can people know how often the data is refreshed or what the common data elements are? You are muted.
Yeah. So we should be getting our first EPIC download today from the CTSI Bioinformatics program. We do have one research project which has been entering information on hospitalized patients, but we're about to start the collection of data from the from the medical record chart abstraction. So beyond the research projects that have already begun contributing data, that process is just beginning. But we should have we should have the data pull from Epic today. In terms, we've developed an intake form, and I'll try and get that link so that we can send out that link to our project intake form, where we ask some questions about study design and study populations, and subsequent to filling out that form, we actually, within that form, we have a download for an Excel file which has the data dictionary for all the data elements that we're collecting. It also describes which ones should be available now and which ones should be available in the future.
So Arash, I think this question might be for you. For those of us who are applying for NIH grants, at what point should we put our request into the prioritization committee?
I think if the requirements of the grant are to have access to biospecimens, then you would probably want to do it before you submit the grant, because there are a multitude of other projects that have already made similar sorts of requests, and part of the responsibility of the prioritization committee is to sort through that. There's not an unlimited supply, and no one can predict the number of patients that we're ultimately going to have on either the outpatient or inpatient side to service the needs of the research projects that are being submitted. So the safe thing to do would be to assess the feasibility. No one wants to go through the effort of submitting a grant and having it funded, but not being able to achieve the goals of what the grant are submitted. And I think it's very difficult to know exactly whether or not there's going to be sufficient biospecimens for all the grants that people are putting in. I will say that there's also a lot of people that are interested in de-identified data and specimens that typically or historically may not be considered human subjects. So we've set up a system with the IRB that if you submit a new application into the IRB that's COVID related, it will flag it so that the prioritization committee and feasibility can be assessed based on the IRB application and the protocol that's submitted to the IRB. It reduces the effort of having to fill a bunch of information on the link that we've provided. But we recognize that a lot of people already have IRB approval and or maybe doing things that traditionally the IRB would not be considering human subjects. So we needed to create a pathway so that they can also be reviewed, so that these questions can be answered, especially if grant applications are going in or LOIs are going in for clinical trial concepts without really knowing what the landscape of those are going to be.
Okay, there's a couple of questions about how we're publicizing trials that are already active, and will there be a central site where people can access what research is already happening clinically?
So, of course, this has been a discussion that went back and forth in some emails today. UCLA Health has a website that lists all the clinical trials, but we should do a better job of making sure that people know how to access that. It was part of the communication plan before COVID went live, but this is an opportunity for us to make sure people know how to find the active clinical trials. The School of Medicine is actively working on a website to highlight the entire breadth of COVID research, including the clinical trials, but a lot of the other projects that may not be specifically clinical trials. And there's a huge team that's working very actively. And in fact, the submission of something through the IRB or through the link that got distributed provides some of the basic information for that team to be able to reach out to investigators and understand what they're doing so that they can appropriately highlight that on the website. And I don't know if Kelsey or Steve or someone wants to speak about the initiatives inside of the Dean's office.
I'd love to. So we have a comms IT group hard at work on developing a universal intake form that will do two things. It will help PI requests get routed to the right committees in a user-friendly way, but it'll also be an intake form that includes project registration that we can use that information to feed into a database, a list of all of the COVID-related research that's going on.
And then Arash, if you could just summarize once more the sequence of events that should happen if an investigator wants to submit both to the prioritization and to the IRB. What the question is the appropriate sequence.
Yeah, so if it has not been submitted to the IRB yet, they should just submit to the IRB. It'll be queued for the prioritization committee to review. If for some reason it's not considered to be human subjects, and the advice of the IRB is that you don't need to submit to the IRB, then you should submit it directly, because still de-identified biospecimens and data may require the infrastructure that we've laid out, and we want to take that into account. For those individuals that are trying to do some feasibility assessment, the link also provides an ability for us to try to be able to do that if that's required prior to an IRB being submitted. But the easiest thing to do for new ideas and concepts that have not been through the IRB is just to submit to the IRB, because it'll queue it up for review without you having to do much else at this point.
Okay, and there are a couple of questions about surveys. What is the pathway for surveys that in which people would like to survey healthcare workers or patients rather than using biospecimens?
Yeah, I think that's a really great question, because you can imagine that there may be dozens of people that want to survey the same group of individuals. And so the challenge is going to be how to make sure that when patients are approached, they're not confused about why they're answering similar questions from different groups that are trying to get them to do the same sets of surveys. So I think we have an obligation to our patient population to do some air traffic control and to get people that are interested in the same topics to collaborate with each other, so our patients only have to answer those questions once in a way that services the needs of the majority of the researchers that are interested in those responses. Symptoms is a great example. Many, many people are interested in the symptoms of these populations, but I don't believe that it needs to be asked by 10 different studies over and over again, because I think it creates a burden for our patient population that I think we have some obligation to try to coordinate.
Okay, and Kelsey, this might be a question relevant for you. What about projects and IRBs which were approved a month ago and papers are already written?
Yeah, that's another good question. So there are a couple things about that. One is we have started what we're calling a pre-review committee that has 5 individuals on it, where we're strongly recommending that those manuscripts be sent through that committee, just to ensure that the data is scientifically sound, and to help accelerate publication. One of the issues there is that it's really important that the IRB, if it was given for prospective studies, that it actually doesn't rely on data that was collected prior to the IRB approval. So I think that it would be useful to have the manuscripts sent to the pre-review committee, and that data is available on the DGSOM COVID-19 website, so that we can determine whether that falls into that correct category. We have had some concerns about issues that Dr. Naeem brought up. When there is information, we want to make sure that it's not going to, if there's small numbers, it won't identify any of our employees, that it really was obtained using, in addition to the IRB approval, the correct data approval, use of the data. Arash, do you want to add anything to that?
No,
if you're still there? Yeah.
I am. I think that, there are plenty of people that are concerned that they have a project that's already in flight, and whether the goal of this process is to disrupt that. I think the answer is no. For studies that
No,
are in
yeah.
flight and that are already happening, they're happening because they're probably feasible. You know, there's plenty
Yeah.
of IRB approved studies that are approved but have been sitting on the sidelines because it's been difficult to figure out how to operationalize them and make them feasible.
Yeah.
And really, that's the goal of this process.
Yep. Thank you.
And Steve or Kelsey, is there a general timeline for restoring research operations that have been deemed non-essential?
I can take that. No, there's unfortunately no timeline at this point. I think anywhere in the country, as I've been, talking to people, and definitely not at UCLA or anywhere in the UC system. I think the good thing is that plans are being created. That we're starting to make plans. I think many universities are starting to make plans of what a ramp up will look like. It won't be just a sudden opening of the gates and everybody back to work. There will be certainly safety measures. I think what people are envisioning is still a major effort to try to keep social distancing within the labs, keep the labs at a very low density. For us, the sooner the better. We're just as eager as everyone to get things get research happening again. So we're trying to push the agenda as much as we can reasonably and safely do that.
Great, thank you. Arash, there's a question about Atlas data and the availability of Atlas data associated with COVID-19 positive, negative, or recovered patients.
Yeah, so I think that the intention is to have there be overlap between the COVID registry and the Atlas program. I think that the Atlas program, as part of that program, requires patients to fill out a universal consent process. But for the COVID registry, we've been able to get a waiver of that from the IRB. So the Atlas program will be the beneficiary of a fair amount of the remnant specimens, as well as having the data already probably in place for a fair number of patients that end up being COVID positive. And I think that the likelihood that there's a fair amount of ability through Atlas to be able to analytically be able to look back from a precision health perspective, I think is the intention of a lot of the work that Dr. Elishoff and Dr. Dreyer are doing in terms of setting up this infrastructure. So I think it doesn't have that at the moment. And so I think it's going to take a bit of time for us to catch up to make sure that the Atlas project has that data as well.
Okay, thank you. Another survey question. If I'm participating in a national survey research that's not necessarily specific to UCLA, but UCLA already has an IRB to participate, does that go through the committee as well?
Yeah, I think if it's not specifically focused on UCLA patients or data that's coming out of Care Connect, and it's a national sort of survey, then it is probably going to be not reviewed, exempted, so that it can quickly get reviewed by the IRB. Everyone realizes the time sensitivity of those national surveys, and every attempt is going to be made to ensure that this is not part of that process, to slow it down or to hinder it, if it doesn't fall into the categories that we discussed during the course of this town hall.
And do we know about how long the pre-review process for the manuscripts, how long that will take?
Yeah, well, you were just asking me that as I was texting the head of that committee, but I know that as soon as I get it, I'll type it on the Q&A, but I know that they're committed to a very rapid turnaround, I think on the order of 48 to 72 hours, but I'll confirm that. Okay. They just met this week on Monday.
Great. And then there is a specific question about the Discovery Data Repository, the DDR dashboard, and its relevance for COVID data. And this may have to do with the refresh rate. I don't know, David, if you could speak to that.
Right. So we haven't set this up within our group. I know that Chris Denny has been working on building a COVID dashboard, and so I think this is something that is currently being worked on.
Great. I think we've covered a lot of the questions here.
So I think that a lot of the infrastructure that's going into support research around this, the plane is being built while we're flying it. I think that we wouldn't be able to be where we are today without tremendous leadership and effort on Dr. Courier's part, Dr. Dry's help, Dr. Elshoff, and a lot of the staff that have been actively involved in either opening studies or providing support to be able to get and lift the sort of research efforts up and running. And of course, none of us, including myself, would be successful without the significant support and leadership that comes from the dean's office and from the health system. Realizing that for many of our patients, the best care that they're going to get is going to be what clinical research can provide them. And so I don't know, Kelsey, or if you or Steve have final comments that you want to provide.
Steve, do you have any? Is he still on? Steve, did you want to, Steve?
Sorry, I just had to, I was answering another question as that was being discussed, sorry.
Okay, sorry. Do you have any final comments? I can make some comments. I do. There are a couple questions that I've looked at that I'd love just to have Sarah, because we have a couple minutes left. When is the antibody testing going to be available through Care Connect? And there's a question about research, using UCLA core labs to do testing, COVID-19 testing at research prices. Can you say anything about research testing and also about the serologic tests in the lab? When are those going to be available? Yeah.
Yes, absolutely. We plan to have antibody testing up in the lab in May. The vendor that we are planning to go with is getting an emergency use authorization from the FDA shortly. And as soon as that comes into place, we'll be able to start offering that in the lab. Regarding offering COVID testing at research prices, I would have to run that through the COVID command structure at the hospital. Obviously, you can understand that the command structure wants to ensure that we have adequate testing for our patients and our healthcare providers. So I would be happy to speak to those people offline who are interested in that to find out what the volume is that you're talking about and to see whether or not we can accommodate that.
Yeah, I think that that's an important issue. And I think that in framing a lot of these issues, there were questions about PPE, about testing that are, we have to do these in areas of prioritization. And the first priority is ensuring that there's adequate PPE, adequate testing in the clinical arena. And then that opens up, because of course the research arena is going to be critical for the next phase of this, but we're marching through the immediate needs at the healthcare level for our healthcare system and for our affiliate healthcare systems. And, in terms of any overarching comments that I would make, I want to just express how grateful I am to everybody, all the panelists on the call, all of the many people who've really come together in what I consider to be just remarkable ways to think about how they can coordinate their efforts in a time when we really do need to prioritize and coordinate. And it's been incredibly inspiring for me to see. And while I recognize that it's a new way of doing business in many regards, I think it's also an incredibly promising way for us to be able to coordinate what we do best to be able to really have an impact in the near future, and hopefully to be able to address any future pandemics. So those would be mine. I don't know, Steve, if you want to add anything.
No, I just think we're really, what's very impressive to me is the group of people that have been helping put this together. I think Arash and Judy Courier and Steve Dubinet, it's been, as a non-clinician myself, that there's so much of this that requires that clinical expertise and organizational expertise. And I think it's been done just extremely well, and I'm really grateful for that.
Kelsey, can I ask a quick question? I think, a lot of what we've been able to do in terms of lifting this has been done with significant support from the Clinical Translational Science Institute. I know that you and other leaders have been actively involved in raising money from donors and other individuals that might be helpful to the institution down the road. Is there something that people can think about if they have access to donors, or is there a way to get people involved, in terms of that sort of effort that might be helpful in terms of floating the research efforts overall beyond what the CTSI has been able to contribute so far?
Yeah, that's a great question. And we have been successful, and I'm anticipating that we'll continue to be more successful in raising funds for this. And part of what has allowed us to be successful is that we have taken a coordinated effort. And so I really want to encourage people, as soon as the registration form comes out next week to register whatever studies you're doing on COVID-19, to do so, because we are using those as a way of determining, sort of putting projects together that we can bring to very large donors. This is really a time when we can come together as a community to approach our donor community to talk about the really big things that we can do, rather than go out individually and to our own sets of donors. So I would, what I would say is I would encourage you to use the intake form to register any kind of a project. Whenever you're submitting a project for approval through the scientific prioritization and feasibility, that's going to feed right in. We'll know what that is. And that really allows us to go out and say, for example, we want to raise money, for example, for the convalescent plasma program, or we want to raise money for clinical trials, and we can look at all the clinical trials that are there. And it's a much more compelling message to be able to bring out. Any other questions or?
Seems like we're good.
Yeah, I think we're good. Thank you. Thank you to all of you. I really have been motivated by everything that's gone on. And again, I think it just takes advantage of the expertise that exists across our campus and really across the school and the health system. And I'm incredibly grateful for it. So continue to move forward and support the activities that our faculty and staff and trainees are engaged in. We will look at the questions and answers that we didn't have time to answer, to see if we can get written answers to those as well. So thank you very much.
Thank you.